fibromyalgia syndrome, or FMS, have an autoimmune condition? That's the question that we are now going to address. Recent research on B-cell receptor sequencing reveals a lot of immune tolerance in FMS patients. These discoveries paved the way for the new immune system-based therapies and diagnostic tests. Mr. President, you will now welcome Professor Andreas Goebel, who will elaborate further on this topic.
Andreas Goebel was born and raised in Wiesbaden, Germany. He was trained in anesthesia and pain medicine in Würzburg, also in Germany, and in Oxford, in the UK. And he studied post-trauma immunology at Harvard Medical School in the USA. He is appointed consultant in pain medicine at the Liverpool Bolton Center in 2007, and then he became appointed senior lecturer at the University of Liverpool. He is now chair of pain medicine at the University of Liverpool and director of the university's Pain Research Institute. And we will listen to his talk.
Thank you very much. Thank you for the invite, Mr. President. It's a lovely area here. It's incredible. I don't think I've been ever in a room like that with this view. So it's a real pleasure to show our results here to you today. So you've already said my background, explained it. So in the New England Journal over 4 months, these are the images in clinical medicine every week, as you know, and you usually you're asked to diagnose these patients in accordance to what you see, and you can visually inspect or do a blood test, tissue assessment, and the effects are often infection, immune problems, environmental problems, although the pathways are not always understood. But if you go into anywhere out there, now the next 50 people, at least a quarter of people around us have conditions which you cannot depict in this way in the New England Journal of Medicine. You can't see anything, but these conditions are the leading cause of life years lived with disabilities. And they come more in the foreground now that we have some ways to treat the prominent infectious diseases and also some of the cancers. And we've called them now, or I've suggested to call them symptom-based disorders, and we've had a vote in the Royal College of Physicians, and that came up first. Other names are persistent somatic symptoms, medically unexplained conditions, functional neurological conditions, but I believe that these are disorders, and this is the best maybe condition. I should have started this talk with apologizing for not talking in French, because my French is not very good. I'm sorry about that. So I continue in English. So this is like the picture we see when we see these patients. Yeah. So it's like a fog. We don't really have any tests that we can. Okay, this shows us what's going on. In the WHO ICD-11, the painful disorders, the painful, if you like, symptom-based disorders, are called chronic primary pain. And I think it's a genius move. I was part of this group, but it wasn't my idea. It puts together the psychological aspects that are always very often present, and the experience of pain, and you might be able to see that. So these are conditions that by definition are associated with at least emotional distress or significant functional limitations, and they're not explained by anything else. So they're not secondary to any other conditions like rheumatoid arthritis or nerve damage. And in the ICD-11 we've got several subgroups, and we have done research in the first 2 subgroups: fibromyalgia, a common condition, and complex regional pain syndrome, a rare condition, used to be called reflex sympathetic dystrophy. But there are many others that belong under these, including irritable bowel syndrome, chronic migraines, chronic primary low back pain, pelvic pains. My research started with a different condition called complex regional pain syndrome. Here, and I have chosen this because initially you can see something. So So I thought, well, people will probably give me more money if I do research on a condition where I actually can see something, because they believe maybe something else could be going on. Yeah. So initially, often the hand is swollen. It's a post-traumatic condition. It's swollen, but it's not the swelling that's problematic for the patients, not the redness, it's the pain. It's extreme pain in the distal extremity. 95% of patients get that after a trauma, which can be a small trauma to the distal limb. After a year or so, the swelling gets better, and about 20% of patients will still have extreme pain. And we established that autoantibodies are probably responsible, or at least contribute to this condition, and I started this when I was in Oxford in 2005, and this research is going on. But we had a volunteer, a healthy volunteer, who gave blood repeatedly, and the blood was always normal when we used them for comparison, yeah, because we also need healthy volunteers, and then they developed and didn't tell us widespread pain, and suddenly the blood results changed, and that made me think, well, maybe fibromyalgia could somehow be also associated with an immune problem. And in fibromyalgia, as you know, is a common condition. In the UK, it was very important that Lady Gaga, Morgan Freeman, and Kirsten Bannister came out and say, I have fibromyalgia. Now, over the last 5 years, there's a lot more acceptance that that even exists, that this is real, and that we need to do something about it. I've chaired the Royal College of Physicians guidelines, the diagnostic guidelines on fibromyalgia, and this is a pictogram from these guidelines. And as we know, this is not just a widespread pain condition. It's associated also with fatigue. It affects people not just with pain, also with fatigue, with poor concentration, with an unrefreshing sleep, and with when we press the skin, and in females certainly this is often painful. A lot of people are also experiencing a lot of pain when they go out into the cold. We've published, after 5 years of research on this paper together with David Anders, Stuart Bevan in King's College, Camilla Svensson in Karolinska, and many mice. And because you're all doctors, I presume, but maybe on the internet there are people that are not doctors. So what is this about? This is not about inflammation per se, okay? This is about something else, about antibodies. Antibodies is something we all have. They bind to structures on the bacteria, for example, and they look similar, the left one and the right, but the left one can only bind to this little feather, and the right one can only bind to this tomahawk. And once they've bound, the binding causes inflammation and destruction of the cell, the target cell, which is the bacterium. But autoantibodies is part of this system. Maybe it's a prize for having such a good system. We also make antibodies against our own body. Some of these have only function-modifying effects. And this is an example from a condition called neuromyotonia. You've got here a peripheral motor neuron. You've got voltage-gated potassium channels, and you need them because when you have an action potential, you need the potassium ions afterwards to go out through these channels in order to stabilize the membrane. If you have these antibodies, then they bind to the voltage-gated potassium channels, and now the ions cannot all get out. And as a consequence, you destabilize the membrane potential, and when you now have an action potential, you get spontaneous potentials and you get involuntary muscle movements. There's virtually no destruction of any tissue, there's no systemic inflammation. It's just this what is happening. So I hypothesized that maybe in these unexplained chronic pains, something like that is going on, something that is affecting the sensory neurons or cells around these sensory neurons. How do you find that out in animals? So firstly, there is a model called in English passive transfer model, where you can take the antibodies from a patient, the good ones and the bad ones, all the antibodies, you take them from the blood, and you purify them and you inject them to mice. And then you observe the mice. And why would that work? It works because the mice are surprisingly similar to us. Okay? So their cell surface structures are quite similar as our cell surface structures in many, many instances at least. And if you're lucky, for example, in myasthenia, which is a muscle condition caused by autoantibodies. If you take the antibodies from a patient, inject that to the mouse, it takes about 2 weeks, and then the mouse will get weak limbs. Patients suffer from weak limbs because the antibodies interfere with the neuromuscular junction. And so we tried this, and how do you do that in a chronic pain condition? You cannot ask the mice. Okay, so this interview technique is not yet developed, so you have to test them. And there's a lot of this debate, how do we test for spontaneous pain? We cannot really fully test for spontaneous pain. You can look at movement, and actually these mice didn't move as much, I'm not going to show that now, but the principal way of looking at them is to look for hypersensitivities. People with fibromyalgia typically have sensitivities to pressure or touch, and you can put the mouse here and you poke with this little poking device slowly, stronger, until the mouse says this is too much. And then it withdraws the paw, and you then measure when does the mouse withdraw the paw. Eventually, the mouse will withdraw the paw always, because if too much pressure, we will withdraw our hand if someone puts too much pressure in there. And we tried this out, and it did work. So we put, you can see here the healthy control injected mice. About 110 grams are needed for a mouse to withdraw the paw, and then if you inject the IgG of the fibromyalgia patients to the mice, then the mouse becomes profoundly hypersensitive. And that we tested a few other things. So the mice lose grip strength. People tell us with fibromyalgia that things fall out of their hand, they don't have the strength. Nerve conduction studies are normal, okay, so you cannot measure that as normal. Microneurography studies are abnormal though, so if you go to a single fiber, you can see that these fibers respond differently. So you can see clinically a problem if you like, but there's virtually no service to do that. So, but anyway, so the mice do get weak. They also get a mild small fiber neuropathology. So the nerve endings in the skin are rarefied, which is something we find in patients, in about 50% of patients. And this happened with all preparations we tested, and all control samples always tested negative. We hadn't expected such a profound result at all. So we would have been very, very, very happy if we had found this in 25% of patients. But they all have severe fibromyalgia. We should also say that they are patients from our pain management program. I work clinically in the pain service, so I also see patients for the pain management program, where they learn how to better move, how to better also psychologically deal with it. So they come only there if they have really severe fibromyalgia. Camilla Svensson, who worked together with us in her lab, found out that there are, because we don't know how does this work now, so why would that happen? Where do the antibodies bind? And she found that they probably don't bind in the brain, they bind certainly in the dorsal ganglia glial cells. You see here a dorsal spinal cord from a mouse cross-section. This is a dorsal horn here. This is a dorsal ganglia. In the dorsal ganglia are all the cell bodies of the sensory nerves. And around these cell bodies, which are these big blobs, these nerve cells, they're big, are little cells. You might be able to see that. And the antibodies bind to these little glial cells. We don't know whether that's what's causing it, but it is kind of suspicious, isn't it? Because they're quite close to sensory nerves, so they could maybe change the function of these nerves. We know that in the animal model, these glial cells are activated. Okay, but this may not be the only way. So we have now, this is under review because there was things on Dong and Johns Hopkins. We looked at mast cells, and you can maybe just see that here there's a lot of red dots in the mice. The mast cells that have been injected, so the mice that have been injected with fibromyalgia IgG, these mast cells increase in number, and you can see here the difference. And then if you take from these mice mast cells, from normal mice, and incubate them with fibromyalgia IgG, you probably cannot see that, sorry, but there's lots of green here. So there seems to be a direct binding of the fibromyalgia IgG to mast cells. And actually, if you knock out the mast cells in the mouse, we've done that as well, then the whole phenotype goes away. And that's, you can look that up, it's now on a pre-pub. In our lab, we've looked at skin of patients with fibromyalgia, and that has already been shown before that patients have more mast cells in their skin. We were able to reproduce this. Look, these are all 3 standard deviations above the normal. But what we found for the first time, you can see this here, it's a bit brownish. It's not just mast cells increase, there's also tryptase that is getting into the tissue. So these mast cells are clearly activated. So therefore, one could say maybe if this is the B cells are clearly needing to be abnormal, and they produce these antibodies which firstly bind to dorsal ganglion cells, which then activate sensory neurons. This is a model, this is a hypothetical model, yeah? And then sensory neurons being activated might cause abnormal signals up to the brain. And sensory neurons being activated will also lead to a peripheral, a release of neuropeptides, including substance P, and maybe they activate mast cells, and additionally there's direct binding to mast cells. So maybe there's sort of a vicious cycle going on. We cannot yet, this is sort of a hypothetical model. So this would all point towards a defect in B-cell tolerance, wouldn't it? So there's something wrong with the B-cell, they shouldn't really produce these antibodies. And we've worked together with Professor Rachel Bashford-Rogers in Oxford, a professor for biochemistry, who developed these and published this in the first author science paper. So we did B-cell receptor sequencing, and we also did flow and we did stimulate these B cells. So we take white cells from patients, look what is in them, are the populations in any way different, and then we stimulate them with T-cell independent factors, and then we look how the flow looks like then. We also look independently how the B-cell receptors look like. I'm just going to show you the little part of this which I understand a bit better. The paper isn't out yet. So the simplest and most graphically interesting part is how these B-cells proliferate. Yeah? So you can see after 5 days, the yellow triangles mean that the fibromyalgia B-cells proliferate more after 5 days when compared with healthy control B-cells, and this is across all subtypes. We think there's a problem with the naive B-cells already, but not with central tolerance, with peripheral tolerance. So Rachel put this together as in this way, and she's normally working with classical rheumatological autoimmune diseases, and there are a lot of similarities. So how does this start? Not everyone gets fibromyalgia, but a lot of our patients, many of patients tell us that their fibromyalgia started after horrible experiences. Okay, so they say, I got divorced, I got dismissed from my job, I got abused, and often on the background of other things that happened to them throughout their lives. So I propose that the experience of severe trauma in genetically predisposed people can produce this loss of B-cell tolerance. We know from long COVID that maybe infection can have a similar effect, and we know if you look at fluoroquinolone toxicity, that looks like fibromyalgia sometimes, so maybe there are other effects how this can be caused, but I think that trauma is a major one. And so maybe they all can cause this, but then recently, if you might want to look this up as well, 4 weeks ago this paper was published by a Canadian-Israeli group showing that the gut microbiome is profoundly abnormal in fibromyalgia. When you transfer that to mice, the mice get fibromyalgia. It's very similar as our mice, but it takes 4 weeks, whereas if you do the antibodies, it takes 2 days, so that this could be very much the same effect. So it could be that the microbiome then primes these B cells to behave differently, and there's actually in our sequencing some hints that that could be the case because IgA is particularly dysregulated. And so maybe how this works is that the stress works on the microbiome. What does this mean for future healthcare? Well, it just means we should maybe, certainly it has stimulated me to get together with the Royal College of Physicians and think about how should we maybe treat these people in the future. Certainly, I think we should have better ways of organizing our healthcare systems so that they don't have to go around to many, many doctors or being dismissed. Maybe we can educate people, maybe we can also create special services. one last slide, if I may, is that this also may have implications for the next generation. In my patients, I started 2 years ago to become alert to this. So people report, I've got 2 sons with autism. It shouldn't really be that frequent. So in my patients, in 500 patients over 2 years, the frequency of reported autism in children was 16%. In other patients, in my other patients with back and leg pains, it was 5%. It should only be 2 to 3% of the general population. So there's many reasons. We have not found really a genetic load in the relatives. This is under preparation now. But many reasons. It could be birth complications. But one reason which we know in autoimmune conditions is that in the 3rd trimester, IgG, abnormal IgG, can go over the placenta barrier and can cause abnormalities. So we wonder whether that could also play a role and cause potentially hyperalgesia and possibly also neurodevelopmental problems. So in conclusions, these disorders are costly and poorly understood. The passive transfer experiment suggests that IgG-related autoimmunity plays a role. The mice really develop surprisingly similar versions of these conditions. We will now check that in functional neurologic disorders as well. We have a service file. We are just trying to get and see what happens whether some of these mice get paralyzed, I don't know. The microbiome is abnormal. And this has implications for future treatment. I haven't shown you what treatments we have done, but I think there will be implications certainly for fibromyalgia, both for diagnosis and for treatment. We need to understand better how distress can cause this. Can we prevent terrible distress? I mean, could that as a society prompt us to have better protective measures in place? And what does this mean for the next generation? Thank you very much.
Thank you very much. What is known about the specificity of those B-cells and antibodies?
Well, the antibodies we only know in fibromyalgia, they do bind more than they should do to the satellite glial cells and to the mast cells. That's what we know so far. It's quite a new field. We don't know the molecular specificity. We are doing immunoprecipitation experiments in this with mass spec to see whether we can find out, but it's not easy. So, yeah, but they seem to activate the mast cells as well, so that's what we know so far. In the B cells, yeah, that will come out soon. There are changes throughout the B cell receptor repertoire. Naive B cells seem to be particularly affected.
Okay, and what is the isotype? It's mainly IgG, that's not.
Yeah. Yeah, so we have injected the serum components without IgG. That was always without effect in our experiments. So far we think IgM is not involved here.
If IgG autoantibodies are responsible for the disease, then immunoglobulin, which is a molecule which is able to cleave IgG and which is currently used in kidney transplantation in hyperimmunized patients, it's effective, should be beneficial.
Okay, yeah. What's the medication called?
Immunoglobulin.
Okay. Okay, so we've done a trial together with UCB and an FcRn receptor inhibitor to downregulate the IgG. And so there's several other options that we can look at, definitely. But also microbiome transfer, just to say that, so you can transfer actually for Clostridium difficile enterocolitis, you can transfer healthy microbiome, and the guys from Israel and Canada are doing a clinical trial right now. That it looks really promising at the moment.
So you've mentioned, so very convincing, thank you. You've mentioned some similarity with long COVID syndrome, for which there have been some SNPs identified in the interferon pathway. So I was wondering whether there was any planned or currently done GWAS in FMS that could identify, genetic predisposition in specific pathways.
Yeah, I think Rachel has done a systematic review of the existing GWAS, and I think interferon comes up there, actually. So there have been GWAS studies, but I'm not an expert on that. Yeah.
Yes, so So I had questions relating to the gut microbiota because there are descriptions of changes of the gut microbiota in autism and so on. And since you see a link in the children, are there similar, the changes in the gut microbiome in autism and this disease?
I think we don't know anything about the microbiome in children of patients with fibromyalgia, but as you know, autism, people who have adult autism have a different microbiome, so that's a different question. So this is all very new, so I don't think we know anything about fibromyalgia children's and relatives microbiome, but I know that the Canadian group wants to look at that as well.
Very interesting talk. So I wondered if you have looked at the role of persistent viral infections in these settings. For example, the ones that are persisting in B-cells that could induce this rupture of Tourette's.
Yeah. So what is causing the abnormality in the B-cells, and could that also be consistent with persistent viral infection? We haven't looked at that, and it's not something I'm experienced in, and I'm not planning to necessarily look at this. My gut feeling is that that's not what it is. So what we did very initially is I'm not showing this in CRPS, we looked at anti-infectious antibodies against between 5 and 10 pathogens. And the tests, if you do it on a group level, they're always abnormal, always. So Yersinia, Enteropolitica, Campylobacter, Chlamydia, Borrelia, they were always abnormal, but there was absolutely no pattern. Okay, and we really looked at that for years, so I don't think that what we found it has anything to do with an infection. I think it's an abnormal reaction of B cells for other reasons. I might be wrong, but yeah.
Thank you very much. I would really love that you are right, because it would be so easier to treat the patients. But as a clinician, I know that fibromyalgia patients are not treated by immunosuppressive treatment or steroids or intravenous immunoglobulins. So how do you explain that? Because
yeah, but you have to look in detail then. So they are not treated, for example, we think that an anti-CD20 antibody rituximab does probably not work, I think. Because otherwise even the rheumatologists, they're not so interested in it, but they would have noticed it, I think.
yeah.
Okay. So plasma cells do persist, as you know, and we also know that general myasthenia, for example, does not necessarily respond very well to anti-CD20 treatment. So that doesn't mean that that isn't antibody-mediated. Steroids don't treat antibody-mediated conditions, even if you have it treats the inflammation that becomes secondary, but if it's just the antibody that functionally changes cells, then the effect is quite minimal. High-dose immunoglobulin reduces IgG levels by about 30%. We don't have...
Parce que je vais causer là. Ça va, je te rappelle tout de suite. Voilà, pas de souci.
On a nog een question sur le domaine. Sorry, we will have a question online, so apparently there's a mix of some.
So we don't know whether that's enough. So I think we clearly haven't done. It's not going to be treated through an anti-inflammatory effect. So the rheumatologist always asks, I don't see that that's going to work. You have to downregulate the IgG production or do something with the B cells directly, but that has to include the plasma cells, I think, and then you don't want to be toxic.
My second question is, those patients very often have other functional disorders.
Yes.
And so, for example, I don't know, chronic fatigue or many neurological bizarre symptoms. How do you reconcile that? Do you think they all have different antibodies against different organs?
Yeah, and so it's too early to say that. We don't know, we haven't got any evidence. The CRPS antibodies that I've shown you a few results are different from the fibromyalgia antibodies, just to say that. Okay, so it's not the same. So if you take CRPS antibodies, inject them to mice, nothing happens. You have to injure the mouse. So I don't know, maybe there is an overlap, maybe there's some secondary effects if you have high pain and you get other problems. No, I don't know.
Thank you.
there is no space for the brain in there. It can be all peripheral.
There's no space for brain. Well, we experience all this in the brain. Of course, we need the brain, and the brain, I think, is also starting this. So when I said, in my opinion, well, I suppose that if the stress is experienced by the brain, okay. We don't know what the effect is then of these antibodies. Could they secondarily work on the brain in that way, just so that if you have this high pain, it works a different way? Mast cells are also in the periventricular spaces, and they're also in the hypothalamus, so areas which should be accessible without much changes in the blood-brain barrier. So we are not there yet.
And my second question related to mast cells, precisely.
Can I finish my answer to your first question? I know I gave the impression I was finished, but I just remembered something. So we are now using the mice in the fMRI, so David Anderson's doing that, and he seems to start seeing similar changes as we see in the patients, but it's too early. But we definitely want to see that, because that's an important question. Sorry.
And about mast cells, the increase of mast cells, are they playing a game? Involved in the pathophysiology, or is it just a consequence, or is this place for 5-HT or histamine? Or?
Yeah. Well, I haven't shown that if you knock out the mast cells in these mice, then the phenotype goes away.
Yeah.
So we think there could be, and also they produce certain substances which could excite sensory neurons. Anyway, even in the bladder, in the lung. So we think this is reasonable to assume they could play a role.
But that's not related to 5-HT or histamine or whatever.
Well, okay, so in the paper we found this is not going through the IgE receptor, it goes through a special receptor which calls the X2 receptor. So we think it's not it's not related to an allergic reaction. It's a different type of pathway of mast cell activation.
Yes, we have a question from a member of the Academy who's online, and this is Professor Delven. Professor Delven?
Oui, merci beaucoup, Vanessa. Thank you very much for your very interesting talk, and I have just a comment. Because I'm working with the team at the Hôpital Universitaire des Enfants Rennes-Fabiola, who is treating children with chronic pain symptoms. And we have observed a large number of patients with neuroatypical functioning, such like high functioning female autistic patients. And there is also a recent publication from the Netherlands on a large number of autistic adult patients, and they have observed an important number of them who have chronic pain symptoms also. So it's in the same way that you have explained before.
Yeah, thank you.
No other question. Thank you very much.